This is the biology exam review for chapters 12 and 13. I'm going to skip the prefixes and suffixes. You can do those on your own. Get a sheet of paper and you can put all your answers down and compare them to my answers. We're going to start with completing the chart, comparing DNA and RNA together. If we do the structure of DNA, that's a double helix. If we do the structure of RNA, two answers here. It's a single strand, and it is also straight. The type of sugar in DNA, type of sugar in deoxyribonucleic acid, is deoxyribose. And then the sugar for RNA, the sugar for ribonucleic acid is ribose. Name the bases. Name the bases of DNA. There's 4 of them. Use the names, not the initials. Adenine, which bonds with thymine, and then cytosine, which bonds with guanine. Now name the bases for RNA. There's 4 of these as well. It's adenine and uracil, and then the cytosine with guanine. Lastly, give the function of DNA. What's the function of DNA? It carries the genetic information. And then the function of RNA is to make protein. You can skip this next section where you have to identify the different types of RNA. We're not doing that, but we will do the fill in the blank, which is blank is the process in which DNA makes a copy of itself. Blank is the process in which DNA makes a copy of itself. That's replication. Blank is a mutation that results in missing information. Blank is a mutation that results in missing information. Deletion. A blank is a change in DNA sequence that affects the genetic information. A blank is a change in DNA sequence that affects the genetic information. Mutation. Number 4, a nucleotide of DNA is composed of 3 parts. Name them. What are the 3 parts of a DNA nucleotide? It's a phosphate, a sugar, and a base. Number 5, during the process of blank, messenger RNA molecules are produced by copying part of the nucleotide sequence of DNA. So what's the process that makes messenger RNA? It's transcription. The combining of amino acids into a protein. The process of putting amino acids together to make a protein. It's called protein synthesis. 2 closely related organisms are bred to have the desired traits and to eliminate the undesired ones in future generations. 2 closely related organisms are bred to have the desired traits and to eliminate the undesired traits in future generations is called inbreeding. Blank uses technology that involves manipulating the DNA of one organism in order to insert DNA of another organism. That's genetic engineering. Replication occurs in the blank. Replication occurs in the blank. Remember, replication is when you make a copy of DNA. So where's DNA located? In the nucleus. Building blocks of proteins are called what? Building blocks of proteins are called what? If you look up at number 6, It's amino acids. Number 11 and 12, what does DNA stand for and what does RNA stand for? DNA, deoxyribonucleic acid. And then RNA is ribonucleic acid. Base pairs are held together by what type of bonds? Base pairs are held together by what type of bonds? Hydrogen. Changes in the DNA sequence of a single gene are called blank mutations. Two possible answers. Changes in the DNA sequence of a single gene are called either gene mutations or point mutations. Proteins are assembled in the blank. Proteins are assembled in the blank, so it's what organelle? The ribosome. And then Ian Vilmot created a sheep named Dolly using what process? What process was used to create Dolly? That's cloning. For the multiple choice, if the structure of DNA were likened to a ladder, what would make up the supporting structure, not the rungs of the ladder? Read those choices. And it's D, sugar phosphate molecules. Which do DNA and RNA have in common? Which do DNA and RNA have in common? They both contain phosphate groups. Letter C, both contain phosphate groups. What occurs during frame shift mutation? What occurs during a frame shift mutation? The answer is D. There's a misreading of the base pairs due to deletion. Number 4. What causes the two sides of the double helix of DNA to stay joined together? What causes the two sides of the double helix of DNA to stay joined together? It's the joining of the base pairs. New inheritable characteristics would likely result from mutations that occur where? New inheritable characteristics would least likely result from mutations that occur, the answer is A, in muscle and skin cells. Which defines a codon in DNA and messenger RNA? A codon in DNA and messenger RNA. It's going to be C, the 3-base code. For number 7, the messenger RNA anticodon for the bases CGT are, find its anticodon, the one that matches it, and your answer is going to be GCA, which is letter B. Transfer RNA is, that's all of the above. It's reusable, it contains uracil, and it transports amino acids. An organism's blank is the total DNA present in the nucleus of each cell. An organism's blank is the total DNA present in the nucleus of each cell. That's its genome. Letter A. That's its genome. Number 10, an electric current is used to separate the DNA fragments according to the size. Is a process called what? And that is gel electrophoresis. Letter B. Blank is a field of study that involves creating and maintaining databases of biological information. Field of study that involves making, creating, and maintaining databases of biological information. That's going to be C, bioinformatics. For the true-false, deletion of genetic information can occur in a gene or in a whole chromosome. That's true. Each replicated double strand of DNA contains an original strand and a new strand. That's true. All mutations are fatal. That's false. The discovery of DNA was serendipitous. Serendipitous means by accident, and that is true. The process by which desired traits of certain plants and animals are selected and passed on to their future generations is called selective breeding. That's true. Griffin is credited with discovering and naming DNA. You don't need to know that one. DNA is a protein. That's false. DNA is a nucleic acid. RNA is an organic compound. That's true. That's the organic compound called nucleic acid. 20 prokaryotes contain DNA in the nucleus. That's false. Prokaryotes don't have a nucleus. And 21 gene mutations are typically more severe than chromosomal mutations. That's false. Chromosomes contain more information, so those are usually worse. For this next section, you can do that on your own. The short answer, why does DNA replicate? It replicates to make an exact copy. How does the ribosome know when to stop assembling amino acids? How does the ribosome know when to stop assembling amino acids? They have what's called a stop codon. What is the function of protein? What is the function of protein? It's for structure and support in muscles. What is the Human Genome Project? What is the Human Genome Project? It is to identify and map all human genes. And then lastly, define genetic engineering. Define genetic engineering. It's going to be any change to the genetic code. Any change to the genetic code by scientists. So not a mutation. Any type of manipulation to the genetic code is genetic engineering. State the function for the following. You'll need messenger RNA, transfer RNA, and ribosomal RNA. State the function for the following. Messenger RNA carries the genetic information. Transfer RNA brings the amino acid to the ribosome, and then ribosomal RNA reads messenger RNA. The last section is making a protein using this particular strand of DNA. This is going to be one of your short answer questions on the exam, and you guys can just refer back to your original test to do that one. And that concludes the review for chapters 12 and 13.